So shall we, sir?
Yes, Reshma. Otherwise, it is getting delayed at the end. Kindly proceed.
Sir,
Yes, Sheila.
நீங்க
Sir,
பேசுறது கேட்கல சார்.
Oh, okay.
மியூட் ஆயிட்டு.
Okay.
நியா சார் நீங்க பேசுறது கேட்கல. ஓகே சார். ஓகே.
so Nia sir, shall we start, sir?
Yes, please proceed, ma. Please
Yes,
proceed.
sir. Respected pattern, co-patterns, esteemed resource person, respected faculty members, research scholars, participants, and my dear colleagues, good evening to one and all. On behalf of the PG and Research Department of Biochemistry, Sacred Heart College, Autonomous, Tirupattur, it's given me an immense pleasure to welcome you all to fourth section of the 5 days online faculty development program on advanced trends and innovations in modern life sciences, organized as a part of our Platinum Jubilee celebration. Now, I request Dr. K. Sheila, Assistant Professor, PG and Research Department of Biochemistry, kindly introduce today's esteemed resource person. We welcome you, ma'am.
Thank you, ma'am. Good evening, one and all present here. We are honored and privileged to introduce a very special resource person with us today to give us the keynote address for Lichens as Biofactories, the frontier of secondary metabolites. That is none other than Dr. S. Senthil Prabhu, Distinguished Professor of Microbiology, Dr.NGP Arts and Science College, Gudiyatham. Sir has more than 2 decades of teaching experience and 15 years of dedicated research expertise. His areas of specialization include microbial bioprospecting, antimicrobial research, and green nanotechnology. Sir has completed postgraduate diplomas in bioinformatics and clinical research and data management, and reflecting his strong interdisciplinary foundation, he has successfully executed several funded research projects supported by UGC, DST SERB, and TNSCST. An accomplished researcher and academician, Dr. S. Senthil Prabhu has published extensively in reputed national and international journals and has contributed significantly through authored books and book chapters. He is recognized research guide, reviewer, academic leader, and his contributions have been acknowledged with numerous honors, including the Best Teacher Award and the Fellowship of the Indian Association of Applied Microbiologists, with extensive experience in organizing national and international conferences, faculty development programs, and also the hands-on training workshops. Such as eminent resource person in with us. Thank you for accepting our invitation, sir. On behalf of PG and Research Department of Biochemistry, cordially invited, sir. Once again, welcome you, sir. Now the session for you, sir.
Yeah, thank you, madam. Thank you very much. Actually, Dr. NGP Arts and Science College is from Coimbatore, madam.
Okay, sir. Okay.
Thank you. My slide is visible to all?
Yes, sir. Yes, sir. Visible, sir.
Thank you, madam. Thank you.
Yes.
And yeah. Good evening to everyone. Members, head of the department, esteemed faculty members, organizing committee, research scholars, and other dear participants and my dear fellow colleagues. It is my great pleasure to be part of this faculty development program organized by PG and Research Department of Biochemistry, Sacred Heart College, Tirupattur. I sincerely thank the organizing committee and the department for this opportunity. And also the nice introduction about me. Thank you all. Today I will be speaking on the topic called as Lichens biofactories, the frontiers of secondary metabolites. Surely this lecture aims to connect classical lichenology with modern biotechnology, genomics, and industrial applications. And now we're just going to enter into the today's topic called Lichens biofactories in frontiers of secondary metabolites. The first slide, which shows that what objectives we are going to discuss today's session. The first one is called as that to understand the lichen as unique symbiotic system, explore the diversity of lichen secondary metabolites, and learn the biosynthetic pathways and biofactory potential, and discuss the applications of these lichen substances in various industries like medicine and environment and etc., and identify the research gaps and future directions about this lichen substances. Now we just enter into the main core thing is called as that lichen. It's a unique organism, unique symbiotic system. What is this lichen? Lichen are a symbiotic association, that is a mutually useful association between the algae and fungi. The algal component is known as phycobiont and the fungal component is mycobiont, and which are autotrophic and heterotrophic respectively. Photobiont are autotrophic and mycobiont are heterotrophic. What the algae will do for this system, the algae prepared food for fungi, and what fungi will do, the fungi will provide shelter and absorb the minerals and protect for their partner. This is called as the unique symbiotic system between the lichen and fungi and algae. And you just this slide, the picture which shows that the lichenization. What is this lichenization? Is nothing but that the uh, this is called as that fungi hyphae. This fungi hyphae is going to mingle with the green color one. The green color part is nothing but the algae. The combination of this association of these two photobiont and mycobiont, finally it forms the particular system thallus. That process name is called as lichenization. And composition of lichen. The lichen is already we discussed about that it is made up of two important partners. One is called as fungus and photobiont. The fungus is called as that mycobiont, and the photobiont is nothing but algae or cyanobacteria. And mycobiont, the fungus, which type of fungus majorly involved in this composition or symbiotic association, especially it's called as that ascomycetes. Majority of the lichen are symbiotic relationship by this ascomycetes and very rarely basidiomycetes. And what type of the photobiont and algae which is involved in this type of symbiotic relationship, that is called as uh, green algae and blue green algae, and especially triboxia. That green algae is called as triboxia, and the blue green algae or cyanobacteria which is involved here, that is called as nostoc. uh, Majorly 70% of lichen is made up of this triboxia. Now we just enter into the growing regions of the lichen where it is growing. Lichen will grow in various parts in the world, especially in the various habitat. In an extreme habitat like lichen will grow in deserts. Lichen will grow in Himalayan regions and rocks, and it will grow in seashores and rocks. It will grow in forest, and also it will grow in Antarctica, and sometime it will grow in metals and rocks and plastics and historical monuments too. These are all the parts where the lichens will available and will grow. Especially the lichens will available in extreme conditions or extreme habitats. And we just now enter into the types of lichen. We divide the lichen into three parts based on the thallus arrangement. That means that body of the lichen. The three type of lichen are there. One is called as crustose lichen, foliose lichen, and fruticose lichen. The crustose lichen is called as uh, just a minute. The thallus form crust-like structure. It is closely adhered to the substrate. That means that family attached with the substrate. This one is called as the crustose lichen, family attached with the substrate. Example for the crustose lichen, Graphis and Fissurina species. And next one is called as the foliose lichen. What is this foliose lichen? Is nothing but that the thallus has leaf-like lobes. They are free from the substrate. It is called as that foliose lichen. The very good examples for this foliose lichen is called as Permelia and Paramotrima species. And finally, very important type of lichen based on the thallus, that is called as fruticose lichen. The thallus of this fruticose lichen are cylindrical or ribbon-like branched. It is called as that old man bird. Example for this fruticose lichen is called as Cladonia and Asnia species. These all type of lichen which is available in India and Tamil Nadu. Now we just enter into the another type of lichen types based on their habitat where it is present. Some lichens are called as that lichenicolous. What is that lichenicolous type of lichen? The lichen which is present in the woods of the plant. And corticolous lichen, what it is says that the lichen which is growing on the bark of trees. And succicolous, these type of lichens living in the stones and rocks. And finally, terricolous. The terricolous means that the lichens which is found in the soil. That means that terrestrial lichen. I am just going to the some of the pictures which shows that the types of lichen based on the habitat. This is called as the lichenicolous, which is growing in the woods, which is a lichen which is we have collected from the Nilgiris. And this one is called as the second one picture is called as the corticolous. That means that the lichen which is growing on bark. Then third one is called as the terricolous, the lichen which is growing in the soil. And this is called as the succicolous, the fourth one, the lichens which is growing on rocks. And you just look at here, this is the lichen growing, lichen, different type of lichen growing in the leaves. This type of lichen based on the type which is called as that follicolous. And the second picture here which shows that ramicolous. What is the ramicolous? The lichen which is growing in the twigs of the plant. So we just discussed in the two types of two different types of lichen. That means based on the thallus and based on the habitat. Now we just entered into the morphology of lichen. That means that cross section of lichen. If you going for the cross section of the lichen, these are all the parts you could observe in the lichen thallus. That means the body of the lichen. The upper part is called as that cortex, upper cortex. The second layer after the upper cortex, it's called as algal layer. The green color one is called as algal layer. The first one is called as fungi, and the third part, this part is called as medulla, or it's called as the middle part, and the final part is called as the lower cortex. This upper cortex, medulla, and lower cortex are uh, filled with fungal partner, that means that hyphae, and the second layer it's filled with algal partner, that is called as that algae. This is the morphology of lichen. When we go for the cross section of the lichen, you could find out that the arrangements of the algae and fungi. And what is that upper layer will do? That it will help for the production of the lichen species from the UV light. And the second layer, that algae will do that photosynthesis, and it will generate the carbohydrate energy for the lichen. And medulla part, this part it's called as that, which is helping for that storage and accumulation of metabolites, lichen metabolites. This part majorly the lichen metabolites which is accumulate in this part. Then lower cortex, which is helping for that lichen attached with the substrate. Now we just enter into the lichen availability in India. The lichens are the significant component of Indian biota. Nearly 2,900 species which is available in India, which is the almost 15% of the world known species. All over the world till now, nearly 20,000 species recorded lichen species throughout the world in different habitats. And where is the main regions or the hotspots of the lichen in India? Like geographic regions where it is present. Majorly lichen are present in Eastern Himalayan region, Western Himalayan region, Western Ghats, Andaman and Nicobar Island, Central Indian region, Eastern Ghats, Deccan Plateau, and etc. These are all the places where you could find out and collect the lichen. And especially Eastern Himalayan and Western Ghats are the richest lichen region, or it's called as the very important biodiversity, lichen diversity hotspots in India. Right, now we just come to the main core session theme of this session. Why the lichen are called as biofactories? You know that the lichens are producing nearly or more than 1,000 unique secondary metabolites, or it's called as lichenic substances. It's very unique. It doesn't produce by any other organism in the world. Nearly it produces 1,000 unique secondary metabolites. Among the different type of metabolites, more than 1,000 unique secondary metabolites, most of the lichen metabolites are synthesized by fungal partner. It's called as that microbiome. Then these compounds are not found in any free living fungi or algae. Only it is present in symbiotic association, and it's continuously synthesis biosynthesis in lichen, and it needs only very low energy for synthesis of this lichen. So it is very unique to secondary metabolites. These are all called as, that's why it's called as biofactories. Lichens are very biofactories. It's producing unique and very novel compounds. Yeah, this slide shows that lichen substances, different type of lichen substances, or it's called as diversity of lichen secondary metabolites. These are all the examples, some of the examples of lichen substances. One is called as dipsides, dipsidones, dipensofuranes, fulvinic derivatives, anthraquinones, and xanthones and terpenoids. Example for tepsides, atenorine and stitic acid is the example for tepsides. Asnic acid is one of the very familiar and very important lichen acid from dipenzofurans, and valpinic acid and perietine from anthraquinone. These are all the very important lichen substances. It has the various activities, biological activities. And now we just come to the another thing is called as how this lichen substances are synthesis. The lichen substances are synthesis through these type of biosynthetic pathways. These are all called as the chemical machinery behind the magic, that means that producing unique secondary metabolites. The pathways are called as polyketide pathway, Sikimate pathway, mevalonate pathway, acetyl malonyl CoA pathway. This polyketide pathway, it is called as very important pathway to produce majority of the lichen substances. It is the source of most lichen acids. It producing that various lichens which we discussed earlier slide. Those type of lichenic substances are producing through only polyketide pathway, and the Sikimate pathway producing aromatic amino acids, mevalonate pathway producing terpenes and sterols. Just look at here. This is the overall view of the synthesis of lichen. How the lichenic substances are synthesis in the lichenic symbiotic relationship. The protobion or algal partner are releasing carbohydrate through photosynthesis. This carbohydrate is taken by the microbiome for primary metabolism. After the primary metabolism, it releases some intermediate, that intermediate called as acetyl CoA and malonyl CoA. These intermediates then enter into the secondary metabolism to produce secondary metabolites through polyketide pathway. Which enzymes are the particular system which is helping for releasing secondary metabolites, that is called as polyketide synthase. This is a group of enzymes which is helping for the releasing of secondary metabolites through cyclization and modification and forming aromatic cyclic carbon, and finally it releases lichen secondary metabolites. So these enzymes, polyketide synthase enzymes, are responsible for producing the lichen secondary metabolites, unique metabolites. And this is the slide and the pathway which shows that polyketide pathway. Already we have discussed acetyl CoA. It's a starter unit. It's intermediate of primary metabolism in lichen, and it is combined with malonyl CoA with the help of PKS. What is a PKS? It's nothing but polyketide synthase. It is entered into the linear polyketide chain, and it forms cyclization through enzymes, and it giving aromatic ring carbon. Finally it forms lichenic substances. Just look at here that overall view of the biosynthetic pathway. Algae and fungus or symbiotic association forms lichen. Then with the help of algae, it producing the sugar. This sugar, it releases some primary metabolic compound. Then it releases some intermediates or precursors. Then it is entered into the various pathways, secondary metabolic pathway. It's called as that acetyl malonyl pathway, malonate pathway, and cyclimate pathway, then producing various lichenic substances. Again, this slide also explains that the pathway, this acetyl polymalonyl pathway, it's very important pathway. Otherwise, it's called as, already we have discussed in the previous slide, that is called as polyketide pathway. It releases various important compounds like dipsidones, and dipensofurans through intermediates like beta arsenic acid. And the malonic acid pathway, which is from acetyl CoA and acetyl CoA, and it releases terpenes and some steroids. These terpenes are lichenic substances. It has the more biological activity. And this fungus, through amino acid pathway, it releases enter into the cyclimic acid pathway. It releases some compounds called as fulvinic acid and its derivatives. That's all about that, the synthesis of lichenic substances and their biosynthetic pathway. Now we just enter into the, already I have explained that PKS or polyketide synthase, very important enzymes. These enzymes plays very important role in producing of secondary metabolites or lichenic substances. It is these enzymes are encoded by particular gene clusters. It's called as the cluster of genes. It's called as PKS gene clusters. It is nothing but the group of physically linked genes. What is the role of these genes which is involved in biosynthesis, modification, regulation, and transport of specific lichen secondary metabolites? By activating this PKS gene clusters, only the lichens could synthesize the lichenic substances. And what is the significance? What are all the significance of this PKS gene clusters? It is the basis for chemical diversity of lichen, and it is useful for chemotaxonomy. And when we go for that synthesis of lichenic substances in the laboratory, we could target for metabolic engineering, and it is very important for drug discovery. And the silent gene clusters, most of the time these lichen substances are silent. We could activate these silent clusters through various genomic approaches. Now we just enter into that identification of lichen. How we could identify the lichen? You know that we are using various methods to identify the bacteria, fungi, and etc. Here also we are having some methodology to identify the lichen. Especially what methodology? The major thing is called as color spot test. We are using various colors like potassium hydroxide and bleach, or it's called as calcium hypochlorite, or both combination Kc and Ck, calcium hypochlorite and potassium hydroxide, iodine and paraphenylene diamine. When we are adding this drop of these chemicals on cortex and medulla on the lichen. Cortex, you know that upper cortex, and medulla is the middle part of the lichen. When we are adding these chemicals in the lichen, it is react with lichenic substances. It will produce some colors. Based on the colors, we could identify the lichen. This is called as color spot test. You could do it in the field itself, or else you bring the lichen, you just make it into the laboratory. Another methodology to identify the lichen. You all know about this type of methodology. It's called as thin layer chromatography. Through RF value retention factor of lichenic substances, you can identify the lichen. We are having standard books for that. And another thing is called as HPLC, high performance liquid chromatography. By using standard, you could identify the lichenic substances like atranorin and examples with using standards. By using the peak value, you could find out the lichenic substances. Now we just enter into the industrial applications of these secondary metabolites, where we are using or where we could apply this type of lichen and secondary metabolites. You know that lichens are natural biofactories of unique secondary metabolites. Already we have discussed. It has various applications on the various industries like pharmaceutical, cosmetics, food, textiles, environment, and green chemistry. We are going to discuss one by one what is the use of these chemicals like lichenic substances, especially the first one, pharmaceutical and biomedical industry. It has the antimicrobial activity. Various lichenic substances are using as antimicrobial agents against bacteria and fungi, and it has the strong anti-TB activity. Some compounds are anti-tubercular compounds, that means anti-TB compounds, and some are has antiviral activity, and especially arsenic acid has the anti-cancer activity. It's the anti-cancer compounds, and it's also giving that anti-inflammatory and analgesic activity, and these are agents for analgesic activity and anti-inflammatory activity. Here it's mentioned some of the key lichen substances like arsenic acid, atranorin, salicylic acid. We are using for various biological activities: antibacterial, anti-TB, inflammatory, and antimicrobial, anti-cancer activity, and etc. And in cosmetic industry, how we are using, and most of the places they are using in cosmetic industry and personal care industry, because it has the UV filters. It is involved in UV filter, natural UV filter, and it contains antioxidant. So they are using as anti-aging agents and preservatives for cosmetics and personal care products. And which compounds, especially some of the examples for compound which is used in cosmetic and personal care industry are arsenic acid and lecnoric acid, where they are mixing with sunscreens, anti-aging creams, and herbal cosmetics. Now we just come to that another important industry, food and nutraceutical industry. You people might observe this type of lichen in supermarkets. These lichens using for ingredient as ingredient in biryani and etc. It is also we are using as antioxidant to mix with the food items, and as a preservative, so you can use the antimicrobial activity. So we can mix with the food to preserve the food items, and some lichens are using in functional food ingredients, and some are fodder. It's a feed for cattle. Many of the places in the world, the people are using this lichen as the fodder. And we just come to the textile and dyeing industry. We could extract the very important dyes or natural dyes from lichen, eco-friendly dyes. It is acting as the biomordant, and we could use it this dye in antimicrobial textile finishes. The example of pigment, one of the pigment is called as arsine, which is from the lichen species, is called as rosella species, and which is involved in making of this paper. You know very well what is this paper, litmus paper, or it's called as pH paper. This paper is coated with this chemical pigment, which is derived from lichen. It is acting as the pH indicator, and you just look at here, this is called as the lichenic dye, which is using in coloring the silks in a textile industry. And what are the role of this lichenic substances in agricultural biotechnology? Some lichenic substances are lichen using as biopesticide, because it has the antifungal agent, plant growth modulation, and pest control agent. So we could use the lichen and lichenic substances in agricultural field also. Very important point here, environmental biotechnology, the role of lichen. You all might have heard about that. Lichens are natural bioindicator. So we could use this lichens or for biomonitoring or bioanalysis of air pollution, especially to check the heavy metal accumulation in the air, and it is acting as ecotoxicological indicator. Especially in industry, they are using environmental impact assessment and green compliances monitoring. This image shows that the researchers are transplanted the lichen in the trees to analyze the air quality. The what is that this study which is indicates that the study shows here in Delhi, they studied that air pollution monitoring in Delhi city by using lichen transplant technique. What is the specialty and what is the use of this lichen as a biomonitoring? It's a very cost effective manner you can analyze the air pollution. And our lichen is very sensitive air pollution. Most of the lichen doesn't grow in our cities and towns because of air pollution and air contamination. And and from since ancient days, lichens using in traditional medicine in Ayurveda, Siddha, and Tibetan medicine for wound healing, respiratory ailments, and etc. Hello, sir. I'm
Yes,
audible.
sir. Yes, sir.
Yes, sir. Audible, sir.
Yeah, thank you.
Audible, sir. Audible,
Yeah,
sir.
thank you. Now we just come to the point, like in as the various activities or potential organism. But what is the problem for mass cultivation or industrial application, mass production? The challenges for lichen cultivation, it has the extremely very slow growth rate. The slow growth rate, that means that nearly it grows zero point 0.5 mm to 5 mm per year, and very difficult to synthesis artificially make the thallus in the laboratory. And it has the complex symbiotic regulation. It needs the symbiotic process to regulate activate the lichen substances uh, production. And what is the problem in conservation issues of lichen and sustainability of lichen in India and all over the world? Over harvesting for various purposes and habitat loss, especially during process of deforestation, etc. And air pollution also causes the conservation issue. And we just concentrate on conservation based on bioprospecting manner. We should utilize for future usage because of lichen contain various secondary metabolites. It is a biofactory. So what we should do that, what are the strategies we should follow to cultivate the lichen in the laboratory? That means that to isolate the microbiome alone as a axinic culture in the laboratory. That means that to isolate the fungi from the lichen to cultivate in the laboratory, or co-culturing in the laboratory. That means that cultivate both photobiome and microbiome, or you just go for bio-based reactor, or sometimes elicitate or stress induction to induce the lichen to produce the lichenic substances. These are called as in vitro production strategies to cultivate the lichen and producing the lichenic substances. And this picture shows that in vitro culture of some lichenic fungi. We could isolate the lichen from fungi from the lichen as a tissue culture manner. Normally we could separate the fungi from the lichen. We could grow in the medium like PDA or SD and laboratory in very careful manner or aseptic manner. And this second plate which shows that isolation of photobiont or lichenic algae. uh, especially that green algae and cyanobacteria. But what is the problem here? The lichen doesn't produces the secondary metabolites in a separate manner. That is the problem. It needs that symbiotic association to induce lichenic compound production through PKS synthesis. Activate the PKS. PKS means that polyketate synthase. It needs some regulations and symbiotic regulations and needs some environmental factors. So what we should do that we should go for the inducing or production of lichen substances. We get solution through some modern approaches. What are the modern approaches? We could go for that isolation or making the production of lichenic secondary metabolites by genome mining. Genome mining, heterologous expression, CRISPR based pathway activation, and synthetic biology approaches. What is the genome mining approaches? How this technique we could use it for that induce the synthesis of lichenic substances in the laboratory or in the field. uh, Mining means that to identify the pks clusters. PKS cluster means the gene clusters, polyketate synthase clusters, by some molecular approaches or bioinformatic approaches by using these type of softwares. Example, PRISM, prediction of informatics for secondary metabolomes, and cluster finder. To finding out the PKS cluster is called as genome mining. After that genome mining, you can isolate the PKS cluster and insert into the various hosts like Aspergillus niger, Streptomyces cerevisiae, Streptomyces, other Streptomyces species, and etc. This is called as heterologous expression. That means that you just genome, after finding out the PKS cluster through genome mining, you just isolate the PKS cluster and insert into the heterolog host and express the PKS cluster to synthesis the lichenic substances. And another thing is CRISPR based pathway. You all know about that CRISPR. Here that this slide shows that most of the time the PKS gene clusters are in silent mode. It needs some induced factors like environment and habitat factors induction or symbiotic signals it needs. That time we just using CRISPR based pathway activation of this PKS clusters. What is that CRISPR based pathway activation? You just prepare guided RNA or gRNA. Along with this guide RNA, you just use this DCAS9. DCAS means that dead gas 9. If this Cas9 doesn't cut or alternate the gene, it is only helping for to activate the promoter region. That's why it's called as DCAS9. It is the transcriptional activator. It's binding with promoter region both with the help of guiding RNA and DCAS9. It is increases the gene transcription and it is activate the biosynthetic pathway. So it is producing secondary metabolites. This is called as CRISPR-based pathway activation. And synthetic biology approaches, you know very well that we just find out the lichen genome and identify the PKS through that genome mining. We take the PKS cluster gene into cloning and into the heterologous and you just look at here and express the clusters into the host. We just synthetically making these genes and express the genes inside the host. That's called as synthetic biology approaches. And now we just come to the another point. Already we have discussed, already we would studied about that lichen not only just the combination of two organisms, fungi and algae. It has the several organisms. It contains the various microorganism. It contains the microbiome that is called as bacteria. Various bacterial genera are present in the lichen. Uh, Other lichenic fungi also, most of the filamentous fungi in yeast also present in the lichen, that is called as endolichenic fungi. The previous one what we have discussed, that is called as lichenized fungi. This is called as endolichenic fungi, which is present in the lichen. It's majorly not involved in symbiotic relationship, and the virus is also present in the lichen. These are all called as endolichenic organisms. It has the various activities, biological activities. These areas are well less studied one, unexplored area. We are taking that research in endolichenic organisms like endolichenic fungi, endolichenic bacteria, endolichenic viruses, and etc. And what are the research gaps which is there in lichenic studies? Lichen biodiversity are unexplored in India. Majority of the lichen biodiversity and lichenic substances, we could go for that from the areas called as Western Ghats, Himalayas, and we just go for the chemical profiling of these lichens. And till now, incomplete understanding of symbiosis. It is contains the microbiome, photobiome, and it is also has the associate microbiome. We could go for the functional studies of what is the role of this multi-partner symbiotic relationship, and lack of integrated omics studies. Very less studies to approaches through genomics, metabolomics, and transcriptomics to find out the various functions of lichen. And very lack of studies in synthetic biology, lack of pharmaceutical validation of lichenic substances like preclinical research, translation research, and etc. Low interdisciplinary integration with lichen studies, like we need to include AI studies, bioinformatics, chemical engineering, and etc. These are all the research gaps we could find out that in lichen studies. That's all about that my presentation. I think that I have completed my part of presentation. Respected faculty members and participants, I hope this session has highlighted how lichens bridge ecology, uh, chemistry, genomics, biotechnology, and research gap, and etc. I once again thank Department of Biochemistry, Sacred Heart College, for organizing this, FDP, for giving me this platform. Sometimes the smallest and slowest organism hold the biggest scientific secrets. Thank you all for your patient listening. I will be happy to take your queries.
Reshma,
Hello, sir.
Yeah, tell me, ma'am.
Sir, I'm audible, sir?
Yeah, it's audible, ma'am.
Yes, sir. Yes, sir. So thank you, sir, for your sharing your expertise and valuable insights throughout the section. Now we open the floor for the query section. Participants are requested to post their questions related to the section in the chat box.
some
Dear
people.
participants, you are requested to post your queries in the chat box.
Sir, I would like to clarify. I'm not well versed about lichens. What
Yes, sir.
about the tree lichens?
Tree lichens, sir. The lichens will grow in all the substrates, sir. It will sometime it will grow in a tree, on bark of the tree, twigs of the tree, and leaf of the tree. And some lichens will grow on the substrates, substratum like rocks, soil, and etc. It only it takes the help from the substrate for holding purpose only, sir. It doesn't take anything from the uh, substrate. You could find out the lichen from trees, on the trees, rocks, metals, etc.
Regarding that poisonous lichens like that.
Yeah, some of the poisonous lichens are there, especially if you take that arsenic acid. It is also the one of the giving adverse effect when we are consuming directly, because it's a, uh hepatoallergic compound. You could not take that directly as a food. You take only topical applications. The people are residing in forest area. They know very well that how to differentiate the food lichen and fodder lichen. Normally, we people are not consuming the lichen. We are taking that lichen from the supermarket for making that biryani. That type of lichen is called as parmotrima. That is not a poisonous one.
Okay, sir. Thank you. Thank you for your clarification.
Thank you, sir. Any other queries, audience, please?
Dear participants, you have any queries, kindly post in chat box.
You can proceed with formal wind up, sir. Most of the things are reached. Another thing is little bit new thing we have learnt.
Yeah.
Uh,
People may ask some basic questions, no problem.
Yes, sir. Thank you, sir. So once
Okay.
again, we are wholeheartedly thanking you, sir, for accepting our invitation and you share your expertise and valuable
Uh, Thank you very much for one and all gathering here and listening my patient listening of my lecture. I think so. It is a new topic
the
for most of
section.
yes,
the
yes.
people.
As
Let
you
us look
pointed
forward
out,
to an enlightening and interesting section for tomorrow.
Yes, yes, as the resource person pointed out, it might be a new topic. We learnt basic things and it might be helpful for we can get the students to do some small projects by using the suggestion ideas and the applications sir has given. Hopefully, it will be highly helpful to the faculty members, those who are listened his speech. Thank you, sir. Thank you.
Thank you very much, sir. Thank you all. Thank you.
Over to Reshma.
Thank you, sir. Let us look forward to an enlightening and enriching section for tomorrow. Thank you. Thank you all.
Thank you all.
Thank
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 you,
you
sir.
all. Thank you,
Thank you,
Reshma.
sir.
Thank you,
Thank you, sir.
resource person. Thank you, sir. Senthil, sir. Thank
Thank
you.
you, sir. Thank
Thank
you
you,
all.
sir.
Thank you. God bless you all. Thank you.
Thank you, sir. Thank you.
Thank you, Siva, sir. Thank you, Sheila, madam.
Thank you, sir. Thank you,
Thank you,
sir.
ma'am. Thank you. Thank you. Thank you all. Govind, sir. Poo, ma'am. Thank you, all of you. Good night to all.